I view my responsibilities as being a member of the CBCF Scientific Advisory Committee as making sure that we utilize these really precious resources that CBCF has provided to further brain cancer research, not only in Australia, but that the impact is felt beyond Australia and on a global scale. I think it's important that the research is unique about what's going on in Australia and how these funds are used, not only, again, to help globally, but also locally, to make sure that there is an impact of these resources locally within Australia. When I think of what drives me to try to find a cure, it really just comes down to being involved in patient care. It really only takes taking care of a patient from the initial diagnosis through death to make one understand the impact, not only on that patient, but on all of the family and caregivers and friends that person has impacted. It lets you understand how precious life is and how brutal brain cancer is, and how it's important for us to do everything we can to find a cure. For me, one of the really interesting areas has to do with early drug development for brain cancer. And it's the process, I think, the important process whereby we evaluate the drugs early on in these phase 1 clinical trials, where we can eke out some understanding of the effect of those drugs. And in order to do that well, we need to combine a mixture of expert imaging following growth patterns. We need to include the biomarker subgroups, identifying which populations seem to do better. It also requires that we monitor and evaluate different exposures of the drug to the tumor and different dosing schedules that might be more helpful in seeing a bigger effect. Also say, we really have one chance when we're evaluating these drugs, and that this is usually during that early development that we have to really try to achieve the biggest effect. And when we understand what the drug is doing, that's important. And that's why a lot of these window of opportunity trials, I think, are so critical in the early evaluation, so that we understand the exposure, but really what is the drug doing not only to the tumor, but the tumor microenvironment. Yeah, as I just said, I really have more of a passion for kind of the early phase evaluations of drugs. But there's something that a platform trial in glioblastoma that is multinational, essentially a global study, looking at different drugs, evaluating them for early benefit or early futility, and being able to move those forward as quickly as possible for our field. It really is because of the design and because of all these features of this study that really made it imperative to kind of get started and to get rolling and to move forward. As I said, although I really do like early stage development, this really, I think, is a fascinating and important late stage clinical development approach that takes a lot of the guesswork out, that is highly efficient, that uses less controls and gets us the answer faster with less money being put in by each of the companies. So for me, this is really a great opportunity to be a part of this and to have really put a lot of my energy into pushing this forward because of the value I think it brings to our field. Currently, we think of our evaluation of brain cancer with kind of one therapy at a time. And it just seems really unlikely that we're going to get a lot of effect from single agent approaches. That doesn't mean we're not going to get some. And the kinds of effects that we might see could be what I would term subclinical, meaning you don't quite see a prolongation in progression-free survival or overall survival, but the drug is definitely doing something. That really is an opportunity for combinations, and that's where our field needs to be able to move towards, is combinations. But they have to be thoughtful combinations. I really worry about the empiric process of combining drugs in patients because there's so many different options. It has to be incredibly thoughtful and has to include a lot of great preclinical data to be able to push us forward and to think about performing combination studies in patients. And really, in order to do this right, we need to have high fidelity models that evaluate the importance of the target, the importance of each drug, and that we eventually get to the point where we get cell kill. A lot of our therapies, I think, kind of stun the tumor temporarily. Maybe that leads to some survival benefit, but we simply have to kill the tumor. And that is not always an approach in research, and I think it's something that we're going to have to engage much more in the near future. The reality is, I don't have any. There really isn't anything else that I have more passion about than trying to cure brain cancer. So this is it. Sounds kind of boring for that to be the case, but I pretty much wake up every day and think about brain cancer. I've been doing that for the last 30 years.